Hello, everyone. Thank you so much for joining this today's incredible talk will be given by Tal Einav. And my name is Sita Awasthi. I'm your host for this webinar. I come to you from University of Pennsylvania Infectious Disease Division. Before I introduce you to Tal, I want you to know about this incredible conference, ISV Congress we have in October in beautiful South African city, Stellenbosch. So if you're considering joining, please do. Here is the link and the detail for the abstract submission and the early bird registration. You can also use the QR code. And if you have an interest in exhibition and sponsorship, those opportunities will be available to you at the conference. If you are a member, please consider renewing your membership. And if you are not, please join ISV. It's an amazing society for young investigators as well as established investigators in the vaccine and immunity field. We like to acknowledge all the support that we get from our industry sponsors, as well as you as a member. Without your membership and sponsorship, we won't be able to run these amazing programs that we are able to. We have incredible social media presence on all the platforms. You can find us, become a member, and many of the programs are rerun on the YouTube channel, so you can also visit us there. Our June programs include a next-gen vaccinologist webinar series, which is usually given by young vaccinologist in the field. So again, if you're a young member, please consider joining ISV and you'll have opportunity to join the next gen vaccinologist group. The next one is a mini symposium on HIV vaccines. We have profound list of speakers. You can take a look. It will be on June 24th. It is free and it is very exciting. So please consider joining.
June 26th.
June 26th. Thank you, Ted. Next seminar, the date to be declared, but it will be on impact of vaccine in reducing antimicrobial resistance, and it will come to us from World Health Organization, Switzerland. And now here is our speaker, the man of the hour, Tal Einav. He has an incredible training from Caltech and Fred Hutch. He combines his expertise in physics, in biology, vaccinology, and by combining all this, he gives us infiltrating information on the human vaccine responses as well as human infections. So give him a hand and listen to him. One more thing that I want to mention is that please use the chat box for question and answer, and he has enough time to answer all your questions.
Thank you very much, Sita. And just to check, everybody can see the slides and can hear me?
Yes.
Okay, wonderful. So thank you again, Sita and Ted. My name is Tal Einav, and my lab is 100% computational. So this talk will be a bit different because we're broadly interested in what happens after infection or vaccination to your B and T cell responses. This talk is going to focus on influenza, where we make a very highly dynamic antibody response that changes every time that we get reinfected or revaccinated with the new strains that are circulating around the world. And we're trying to predict this process in terms of the bottom half of the slide to see what happens to your response every time that you see a new virus. So for example, the next time that you'll get your flu vaccine, can we use your current state right now, pre-vaccination, to predict what you will look like post-vaccination in terms of your antibody or cellular or other types of responses? My lab has traditionally focused on antibodies solely, but we're rapidly expanding that viewpoint, and we're codifying this expansion through a competition that we're hosting next year, which is done with my colleagues Bjorn Peters, also at LGI, and Steve Kleinstein at Yale, where we're going to take the same 40 people and we're going to vaccinate them year after year after year. And we're going to ask, what does their response look like over time in terms of their genetics, their BCR, TCR, and RNA sequencing at multiple time points, their cytokines to get some innate immunity, and their T cell as well as their B cell responses. And so the challenge that we're going to offer to the community at large is, can you use the day zero data only, which is all that we'll post for the competition, and predict what happens post-vaccination? So everything in purple for these individuals. We set up this competition specifically so that groups with different expertise can all come in and weigh in on the same data set and try to predict the same thing, so that we can finally compare all these different models that we have head to head, see which ones are the best, and whether the weaknesses of one approach can be complemented by the strengths of another one. I'm especially excited that we have all these different time points so we can see the dynamics of what happens post-vaccination, especially this long-term time point that often isn't investigated. And I won't get into it too much, but things such as the antibody responses will be assessed not just against the vaccine strain, but also against multiple different variants. And I'll talk in just a few slides about why that's so important in terms of this prediction challenge. So I invite you all to participate. The competition starts in about 1 year, which is plenty of runway. And the more innovative approaches that we have, the better that this competition will be. As an added bonus, anybody who submits a model for this competition automatically becomes a co-author as part of our paper that we're going to write about what we find out. But more than anything, I hope that you'll just join us to push the field forward and to see what is the best predictive model that we can come up with collectively in order to predict these aspects of the immune system. So we're now gathering data sets to serve as training data for the first year of this competition. As you all undoubtedly know from your own work, posting these data sets as well as collecting them, contacting the authors, harmonizing them, it takes real work. And I am incredibly grateful for Bjorn's team for spearheading these efforts. And today I'm going to talk a little bit about the insights that we've already gained in terms of serology from the dozens of data sets that we've already put together. So to start with, I want to just give you the lay of the land for influenza. Here is what the vaccine responses or the vaccine strains look like from the past 8 years, just for H3N2, which is what I will be focusing on in this talk, but there's also H1N1 and influenza B hanging in the background that do their own, that have their own patterns post-vaccination. And our goal is always to predict what happens in a new season forward in time, which means if we're trying to predict in 2018, then we'll use data from 2017 and beforehand. And then we can use data from both seasons to predict 2019 and on and on and on. Each time we have more training data, but we're also trying to predict something newer. So it might be an easier challenge in terms of more data, but it might also be harder in terms of you're hitting a moving target. So to do this, we take data from our collaborators, all these wonderful people up here, and let me just take a quick 30 seconds to say, please be like these superheroes, and whenever you publish your own studies, please also post your own data sets. Because nowadays, just having a data availability statement saying, come talk to us, send us an email, and we'll send you the data is a surprisingly large hurdle. So take the extra time and with the paper itself, just post the source data. I guarantee you it'll make it so that not only is the paper more impactful, but your hard-won data that you're spending your time and effort getting is going to be used that way rather than lost in the annals of time, which is what we find often happens with data availability statements. And if you post your data, then go ahead and just send us an email and ask, what can you do with this data set? We love to ask questions such as, how similar are your data sets to everything else that we've seen out there? We have now studies from children, adults, different geographic locations, different vaccine histories, and we automatically calculate the distance to each of those different data sets to your own study. So we can ask which parts of your study are universal and which parts are data set specific to just what you see. And the second question we ask is, what types of new predictions can we make with just your study? What new nuances can we find out? It's a more specialized question, but it's what it's really our bread and butter. All right, so with the data that we have, we're going to ask, how well can we do these types of predictions forward in time to a new season? And I'm going to show you our results with 3 vignettes that start with, first of all, what types of phenotypes do we see in the data whenever we look at these responses. I'll go into how accurately we can predict individual responses across studies. And then I'll finally end by just giving you a glimpse into what we're doing now to both unify and massively expand these data sets that we're starting to get for these competitions. Okay, so starting with the first vignette. When you get an influenza vaccine, most of us have some kind of model, some schematic in our head for what that means, for what we expect should happen in terms of different aspects of the immune system. When it comes to the antibody response, if you ask enough people, you typically hear that the antibody response over time is going to go up, it's going to peak, it's going to come back down. I'm going to talk mainly about hemagglutination inhibition, or HAI, which is how effectively your antibody response can inhibit the blood from binding, the virus from binding to red blood cells. So higher titers mean a stronger antibody response, and fold change means relative to pre-vaccination, how much have you increased because of the vaccine. So you really want for this thing to go up as much as possible and stay up for as long as possible. And with the schematic that we have, it becomes more and more useful when you start to put concrete numbers on it. So doing that, the peak response is reached for influenza by 28 days post-vaccination. That's very clear from the literature. And you should ask yourself, well, what does that mean? How big is the peak that we expect to get? So the next time that you get a vaccine, what are you expecting will happen? Is it going to be a 1x fold change where you don't go anywhere? A 4x fold change, which is what we consider to be the minimal appreciable vaccine response. 16x is very strong, 64 and above, you're a monster. So on average, if we look across the US for any sizable population, we see over and over and over again that the answer is that the fold change at its peak is 3x. So there is a response, there's also room for improvement here. And this response lasts for around 6 months before you get back to baseline. All of these numbers are accurate, and they're what you will see in different publications, but they're also somewhat misleading because they are cohort level averages, and there's huge variability in responses from person to person. And that really, it turns out to matter. So let me show you a little bit of a glimpse of how we look at these data sets. We start by just asking about this day 28 peak response and what happens season after season as you look at many different individuals. And it turns out that the antibody response is very constant over these time periods. This is 8 different years, and you can see the median response is either 2x or at most 4x in individuals. We're talking about hundreds of people here each season, so these statistics are solid. And when you see this plot, some people go and naturally ask, well, what's happening in years like 2022 when we get a slightly larger response? It's a totally valid question because the vaccine strain is changing year after year. But what we find more interesting is not the season-to-season variability, which is actually very tight, but rather what happens in any single one of these seasons where there's this huge spread and what individuals do. There are always in each season folks who have no response at all. This is the 1x or lower responses here at the very bottom. And there are always folks who have a 32x or stronger response, which is a very, very strong antibody response. And so I think that this here is a very strong signal that we want to go after, more so even than what happens season to season. So in our analyses, we actually tend to group all the data sets together from all the different years and to power our analyses and ask what happens for the different individuals in these studies. And so I'm going to show you now what the response looks like, not just at day 28, but at the full curve. And what happens whenever we look at individual responses, the over 1,000 responses that we have here, and what kind of phenotypes do we see? So this is what my student Aaron Lane recently did. He looked over time, he asked what the full change looks like, and we found 3 different types of responses. The folks who, as you can expect, showed a very small response. So they bump up a little bit and then just return back to baseline. Almost nothing happens to them. The folks who mount a more sizable response, and now you can see clearly at 28 days post-vaccination, there is a response that then drips down to pretty much baseline by 365 days post-vaccination. And then there are the folks who have a really strong response that maintains this strength out to 1 year post-vaccination as far as we've measured thus far, which is very interesting. So just dissecting these a little bit more, the response in each of these categories starts by day 7. It increases and it hits its peak by day 10, stays at that peak until day 75, and then it starts to decline, either plateauing or going back to baseline. We're the least certain about this one, so we've drawn it as a dashed line, but regardless of whether it goes down or it goes up, the message here is clear because the solid lines are much more solidly fixed. We know that, for example, most individuals across all of these different seasons have this weak response that we're defining here as less than a 4x response at any time. So there's clearly room for the vaccine to induce a stronger antibody response. But of the people that do mount a strong response, it lasts for a surprisingly long time. It's not this peak that comes up and then immediately down, but we see a plateau that lasts for around 2 months at the top before you start to decay, and the decay takes its own time as well. These phenotypes are highly robust and very predictable. If we know just from a single measurement at day 7 what your antibody response looks like, then we can predict it at day 28, 90, 180, 365. We can predict the whole pattern just by how large you are before you even reach the peak, which is in line with what system vaccinology has shown and suggested. And what this suggests to us is that there's these thresholds of the immune response that are dictated and determined very early on in the response. Some people get the vaccine and they say, okay, but I'm not really gonna rev the engine here. Other people definitely start to have a response, something happens, and then the response turns off. And we say turns off because this decay here back to baseline has the same half-life as what happens when you put in a therapeutic antibody inside of a person. So the antibody production definitely begins, and it absolutely seems to stop, and then antibodies just decay naturally. Whereas in durable responders, there's an even stronger threshold that is reached, and because of that, the antibody production turns on and seems to stay on, because there's no appreciable decay that we see here for this past 3/4 of the year. So this was fun, and seeing these phenotypes was interesting, but we wanted to know whether we could push this prediction back so that we don't even need to know what your response is at day 7, but rather we could take it at day 0. Before you even get a vaccine, can we predict what your vaccine response will look like? And that's one of the grand challenges in the field at the moment because specifically of this variability. How do you know if somebody's going to be a weak or a transient or a durable responder? So we set out to do that question, which is predict how people will respond to the vaccine a priori before they get the vaccine at day 0. And the first step that a model needs to have is to decide what the input is going to be. What features are you going to use in order to describe individuals and model their vaccine response? The literature offers many different factors that affect the antibody response. These vary from demographics to what you have received before in terms of other vaccinations or infections, to what is your baseline antibody response against the vaccine strain. So how well do your antibodies target what we're about to put into you dictates a lot about what your antibody response will be post-vaccination. We know that one's important. There's also things like genetics, there's where you are, there is what you receive, and many other health factors. Among all of these traits, as I'll show in just a second, the most important ones that we found were what is your antibody response against the vaccine strain itself? So pre-vaccination, what is your response against the vaccine? And the second most important feature was pre-vaccination, what is your antibody response against other influenza variants? For example, what is your antibody response not just to this year's vaccine strain, but to last year's and the year before that and the year before that? So the way that we found these important features is we simply asked unbiasedly, what does the data show us? And we again did the same thing as before. We grouped all of our data sets together and we said as a baseline, how do people's antibody responses compare? So if you can think about every possible pair of individuals in our study, doesn't matter who they are, what is the similarity between their antibody response post-vaccination? And the answer turns out to be around sevenfold. So one person will have a response, another one can have a response that's up to 7.4x lower, 7.4x higher. On average, this is a huge span of about 50-fold that they can vary across. If you, on the other hand, ask, well, what if we restrict our pairs to just the ones that we think should be more similar, then you can ask which features make people have the most similar vaccine responses post-vaccination. So I like to think about this as a physicist, which is what my background is. So in physics, you have, for example, ideal gases, where if you know the pressure, volume, and temperature of an ideal gas, then you know everything about it. It doesn't matter what shape it's in, it doesn't matter what it's composed of. Pressure, volume, temperature are the knobs that you need, and then you can predict what any experiment will do. So what are the equivalent knobs here for the human immune system? And it might be, for example, the age of people pre-vaccination. So if two people match exactly in their age, will their post-vaccination antibody response tend to be more similar? And the answer turns out to be yes, slightly. So the difference between their post-vaccination responses drops down to about sixfold. And the same thing happens if we ask about sex. So now no longer allowing pairs of opposite sexes, but just requiring female to female or male to male. Does that restrict the post-vaccination response? And the answer turns out to be around sixfold. As we see if we look at the vaccine dose that people receive or their geographic location. So there's some improvement in predictive capacity, but it's not huge. And the largest drop that we found for any single feature was what is a person's vaccine response against pre-vaccination against the vaccine strain. So that's what's shown here. This point on the very left is showing you if two people match pre-vaccination in terms of their HAI against the vaccine strain alone, will they match post-vaccination? And the answer now drops to around 4x. No matter whether you have the pre-vaccination HAI or you add to it age, sex, dose, geographic location, you can see a small drop here, but generally speaking, these points are all at the same 4x level, which shows you that pre-vaccination serology certainly can give you an indication of what happens post-vaccination. There's still this big spread in values. And the biggest further drop that we found was what happens if you expand to not just the vaccine strain by itself, but also to other influenza variants. That's what happens here with this left point. And then again, adding age, sex, dose, or geographic location varies you just by a little bit. But in general, this was the biggest dip that we can found consistently for any other feature that you can add on. So what this means is that we think about people's pre-vaccination response, not just in terms of the vaccine strain, which I'm showing to the right here with this pink halo, but also in terms of what is their antibody response to another virus that happened, let's say, the year beforehand. So each of these viruses can be thought of as the vaccine strain to this year, last year, the year before that, the year before that, and this makes a landscape of the immune response that people have here, what they've seen, and what the breadth of the response looks like. And just to show in another way how important these HAIs are against many different viruses, if we look at different pairs of individuals, then the correlation between their pre-vaccination antibody responses is directly linked to how accurately their post-vaccination responses will be. So the more and more correlated, the more similar two people will look pre-vaccination, the more similar they will look post-vaccination. And the more dissimilar their post-vaccination response will be. So the conclusion here was that we should be able to predict what someone's vaccine response will be as long as we have their pre-vaccination immunity, provided that we measure these critical features, the antibody responses against multiple different influenza variants. I'm going to show in a second what that looks like in terms of the predictions, but just if you're wondering why these particular features matter, we think that it gives you an estimation of what a person's exposure history has been. So for this individual right here, each of these peaks can be thought of as when did they get some type of infection. And Adam Kucharski showed in a really nice paper back in 2018 that the peaks roughly correspond to a person's infection history. You can imagine that somebody else comes in with a very different history, like a very serious infection that happened a few years ago. And notice that both of these individuals against the vaccine strain alone, on the very right, have the exact same pre-vaccination titer of 80. But only whenever you measure these other influenza variants do you see the difference between their pre-vaccination state, and that's going to influence their post-vaccination response as well. These graphs also give you a sense that you don't need that many variants. We found that going down to as few as just 5 viruses gives you a strong prediction of what the post-vaccination response will look like. So that's what we set out to do next. So we asked ourselves, can we predict the responses in a new influenza season? So again, going forward in time, using all the prior seasons for training, can we predict what happens in this current upcoming season? The input for our model tends to be the pre-vaccination antibody responses against multiple viruses. They are often vaccine strains, but they don't have to be. Pretty much any types of viruses work because any types of viruses break this degeneracy of showing you this exposure history. And then the prediction that we're going to make is what's going to be the response to each of these variants post-vaccination. Here are 4 individuals that I'm showing you from 2017, pre-vaccination only. And rather than directly showing you the post-vaccination responses, I like to first show you both what I think and what the model thinks the results will be for each of these 4 people. So starting with the top left, my view of the vaccine response is that there will be some kind of effect for the vaccine strain, which we've now seen is around threefold. And then this response will taper off as you go back and back in time, eventually hugging the pre-vaccination curve exactly. And I don't know exactly if the post-vaccination and pre-vaccination will overlap entirely, or if there will be a small bump. I don't know if the post-vaccination is going to follow the jaggedness of the pre-vaccination or if it'll smooth it out. But roughly speaking, this is the schematic in my head for what I expect from the vaccine response in this case. In the top right example, where the pre-vaccination titers are lower, I expect for the post-vaccination response to be higher because there is this antibody ceiling effect that tells us the lower you start, the higher you tend to end up post-vaccination. And so I know that I'm going to put in this stronger response here on the top right and on in each of these cases. I'll then let the model take its own crack at guessing what should happen post-vaccination. It's trained on 2016 and before data, and it says that in 2017, it predicts that in the top left, the same thing that I said would happen, which is always gratifying. In the top right, it doesn't know about the antibody ceiling effect, so it actually predicts a lower antibody response that more closely hugs the curve, and on in all these different cases. And only once I have this written down do I like to look at the data and ask, well, what actually happens? And in cases where the model and I agree, like in the top left, the data post-vaccination tends to fall exactly where we expect. I'm showing you the error between the predictions of the model and the actual measurements, so between these green dots and the green line every time. And the HAI assay that we're looking at has an intrinsic error of about twofold. So any prediction error around 2x is going to be considered a win for us. That's as good as predictions can be. Anything larger than that, above 4x, is a bad prediction. 2 to 4 is mediocre. So in this top left case, both the model and I predicted a nice response, and we saw that occurring in the post-vaccination data. In the top right case, where I go up and the model goes down and we disagree, the data generally tends to show that the model is correct, shockingly enough, which is both a good and a bad thing. It's like akin to when your child starts to do better than you at sports. You're both very excited for them and also kind of slightly embarrassed. So I'm glad for the model and I'm glad that it works. But to me, what this really shows is that there's probably something that we can learn about these antibody ceiling effects that are much more nuanced than what we think at the moment. And that's what the model is really capturing here. These situations happen in 90% of the subjects that we look at, where the model just accurately predicts what we see with basically twofold error, as good as possible. But there are also 10% of cases that I like to keep front and center in my mind that are simulated by these bottom cases here, these two subjects. This is all real data, where we either massively overpredict or massively underpredict what happens. And we think these are tied to cases where somebody either recently got an infection just before they got vaccinated, or where they recently they got their vaccine and then between their pre and post vaccination measurement, they got an infection as well. We know that that happens in some cases. The fraction of these errors tends to match that, but we're currently doing a study to really solidify that and show that it's true. But nonetheless, even if we go with the 90% of cases that are these total victories, the predictions are very accurate, and it shows that the model is latching onto some kind of key features. I want to show you the results a bit more broadly because we have thousands of individuals. And rather than showing per person responses like this, I want to switch and show per virus responses against all of these individuals. So I'm going to start by the very oldest virus on the left here, and I'm going to show with one data point the difference between the measured and the predicted values for this virus. And that's going to be 1 point. Another person will be another point, and another point, another point. And here is what's going to look like for all of the different individuals. Perfect theory experiment match is going to line up on this diagonal line, and the actual data ends up falling just on this diagonal line. And it does so for pretty much every virus that we look at, where we're showing that we can predict the low antibody responses and the high antibody responses in the full range very accurately. Although in some viruses we do see a tapering off that we know about now and we fix whenever we have more data sets, so in later on years. Even this slide where I'm showing you all the data for these 6 viruses isn't enough to show all the data that we have in a single study because we have 17 different viruses in this specific 2017 data set. So just to show you all of them, I'll show for each of the viruses its average prediction error across all the different individuals, which looks like this, where most of the viruses are predicted with this 2x error. So this is the total victory line for us. And then the final 3 troublemakers are predicted with 3x error, which we still consider to be good predictions, but we would obviously love them to be better. Nothing here is outside the 4x window that we would consider to be poor predictions. And when we look across all studies that we have, on average, our prediction error is rock solid at 2.3x across all these different influenza seasons, showing that we can robustly predict over and over and over again what's going to happen to different individuals as they get their flu vaccine. A really fun part of this paper is that we went in and dug into what the model is latching onto. and how does it explain the variability, the differences between the weakest and the strongest responses. And the paper just came out, so I won't belabor this point, but I'll just briefly mention that the feature that separates the strong responders from the weak responders tends to be the jaggedness of the antibody landscapes. So folks whose HAI looks very jagged, where they are high for some viruses, low for others as they go forward in time, tend to mount very strong responses, whereas folks who have more flat antibody landscapes tend to have much weaker responses. And we're digging into the mechanism of why that happens right now. Okay, so this was very gratifying news for us. We liked the fact that we could predict antibody responses. And so we're now starting to think about what's going to happen with other facets of the immune system when we look at cellular immunity or cytokines or on and on. And so coming back to the prediction challenge that we're doing, we're starting to make some functions now that were going to be very useful for this competition, both for our own modeling efforts and for everybody who participates in the competition. Everything that we make from now on in the lab is going to not just focus on antibody responses, but must be flexible to accommodate other modalities. So at the very minimum, it should accommodate numeric things such as T cell or cytokine values. And we're also working on the sequencing and on the genetic aspects as well, which come in with their own fun, nuanced models. So just to give you a glimpse as to what we're doing, we're asking questions now about dataset similarity. So if we imagine that we have two different studies, they're going to measure somewhat different things. It's almost inevitable with influenza. You might have two studies in different seasons, so they measure different vaccine strain. Even if they measure past variants, like I showed you in the previous studies, which specific variants they measure tends to differ. If you measure T cell responses, you can have different assays, different stimulation, different outputs that you measure. Cytokines, you can imagine different panels that might partially overlap, but not perfectly. And so the two questions that we want to know are, first of all, when we have overlapping features, that's what I'm showing here in blue, across two different studies, how similar are these data sets going to look like? Can we make a general function that takes this overlap and says, these two data sets have high, medium, low similarity, and really quantify that with numbers? And the second question is, if we know which data sets are similar, highly similar to one another, can we then predict what these features would have looked like collectively in both studies? So what the green features would have looked like if they would have been measured in dataset 1, and what the gray features would have looked like if they would have been measured here in dataset 2. And so those are the questions that we're starting to ask with these methods. I'm going to show you some quick initial responses, which of course are going to be just with our serology studies. So everything here is variant data, but we're now moving on to T cell and cytokine stuff as well. And so this is my student Sierra who asked this very question. She asked, what happens when we link these two data sets and think about them collectively as a one single data set? And you can imagine adding data sets 3, 4, 5, 6, up to 100, everything that we can collect from the field. And although the ultimate goal is to predict what these boxes in red look like, these diagonals, so what the green viruses would have looked like in dataset in the top and the gray in the bottom, we start off by asking what happens with the blue features that were measured in both studies. Specifically, can we take one of these features, entirely withhold it, so pretend like this particular variant was not measured in the study, predict its values back, and see how well we did. And if we do this for every one of these overlapping features in every one of the data sets, then we can get a sense of how similar are data sets 1 and 2. And we do the same thing whenever we have data sets 3, 4, 5, 6. We ask pairwise, how similar is every data set? And then just skipping ahead for time, then we ask with the most similar data sets, can we now predict exactly these values that we've withheld one at a time? So here's what those predictions look like. This is one of my favorite schemes because you not only get to predict the values in these studies, you also get to predict their errors. So in this case, from these 7 studies that are spanning human and ferret, vaccination and infection, so very, very different study designs, we can predict one at a time each of the viruses in each of these studies with what we suspect and estimate from our model should be twofold error. So that's what we say at the very beginning. The predictions that we give you should have 2x error on average. And then when we unveil the measurements and ask how well we did, we see that, for example, for these weakest antibody responses, when a measurement was 5, our predictions tend to be around 5 is the most common value, 10 or lower is very frequent, and then everything above that is this tail end of poor predictions where we predicted too high. But the majority of values are predicted spot on, and as I show you larger and larger measured values, the predictions go higher and higher in the diagonal, as they should, with the true error being at the end of the day twofold, exactly what we expected that it should be. And we don't just get these predictions, across the different studies, we also get a sense of which of these studies are the most similar to one another. And then we can step back and ask, what does matter in terms of dataset to dataset variability? Which datasets talk to each other the most? Which datasets talk to each other the least and are off in their own, little island? And so here's this final data slide that I'll show, which shows 3 nice surprises that we found. The first one is that, as you might imagine, if you want to predict a vaccine dataset, you should use other vaccine studies. That's what's shown here in light blue. And similarly, if you want to predict an infection dataset, then training on infection studies is going to be more accurate. But what I would offer you is don't just necessarily be distracted by, huge p-values, especially whenever the number of points gets very large. It's very easy to have large p-values for small effects. The absolute size of this effect turns out to be very small. And my takeaway from this plot is actually that infection studies can predict vaccine studies very accurately. This is around 2.5-fold error here if you purposefully mismatch them. Similarly, if you use a vaccine study to predict what happens in infection data sets, it's again around 2.5-fold error, which is very accurate. So I myself would not be disappointed at all to use an infection study to predict what happens in a vaccination context or vice versa. Age also has an effect, but it's small and subtle, as I alluded to previously. So again, adults are the best at predicting adult data. Mixed data sets that have adult and children data are slightly worse. Children data are the worst. And same thing on the other end. Children predict children data the best. Adults predicted the worst. Mixed is in the middle. Same thing with predicting mixed data sets. But again, the size of the effect tends to be very small. Nothing here is breaking above 3x in terms of median predictions, which is really nice. So even these children studies, when you purposefully try to use children studies to predict adult data, you still do very well. Same thing with using adult data to predict children studies, still this 2.5-fold response. The biggest effect that we found across all the different data sets occurred whenever we looked at timing. And so when you do your study matters because immune responses do change year after year. And when you get out to beyond 10 years, there is a sizable effect. So this is now showing what happens if you try to predict a data set in the late 90s using other data sets from the 90s, early 2000s, 2010s, or on and on, 2020s. And it turns out that this is where you start to see effects, specifically when you use very old studies to predict things that happened 10 or more years in the future. You start to get larger towards 4x error in terms of predictions here. Still not a tremendously bad, I mean, some of these are truly terrible, but on average, you get around 3 to 4x responses if you go out beyond 10 years. If you stay under 10 years, then pretty much it doesn't matter what you try to do. Even if you're trying to make the prediction error as large as possible, as long as you stay within 10 years, more data helps you out. And when you have enough data sets, the predictions just tend to naturally go almost by themselves towards the actual response in whatever you see. So This is great news for us because circling back to the very beginning, it means that when we're doing this competition, that we're now gathering for this challenge as many different data sets as we can from the last 10 years, because all of those should inform what's going to happen in this coming season when we vaccinate people in 2025. And I'm now confident that we can predict what's going to happen in terms of the antibody response. I suspect that all of these other features are also going to be predictable, but I very much hope that you will join us and lend your expertise and enthusiasm to this endeavor, and that when you think about your own data sets, you don't just characterize the data, but try to predict it a priori and see how well you can do. And with that, I'd like to thank you all very much, and I'm happy to take any questions.
Well, thank you so much for this amazing talk. I see a few questions, and I want to remind audience they have an ability to raise their hand as well, and if they want to ask a live question. But I'll read you from the chat box. So the question was asked from Maya. And the question was that to what extent do mouse studies recapitulate these trends, the 10 days and 20 days, etc.? Um, um, and then I think she continues on saying that at equivalent dose and the dosing regimen, do mice recapitulate the 3x titer generally, or do they under or over promise?
Okay, great. So good question. We actually, embarrassingly, do not have mouse studies yet. We do have ferrets. And so I'll answer for the ferret studies. The short answer is that ferrets are different. They also tend to get infected and not vaccinated. And when they do, their responses tend to be higher, but still somewhat predictive of the human response. So unfortunately, that's pretty much where my answer has to stop at this point. To say that there are some similarities, there are some clear differences, and we are currently exploring the hypothesis that if you pre-infect mice or ferrets with an influenza strain, then they tend to become more human-like. That's what we've started to see. I don't yet know which variants you should infect them with to make them human-like, and I don't know if it's different answers for different age groups or something like that, but um, it's I just don't have a good answer for if you give mice the same vaccine that you give humans, will they have this 3x response?
That's great. Thank you. There is another question from Ben Lorman, and he says, great talk. Could you please comment on neutralizing antibody titer versus HAI in terms of correlation for the prediction?
Okay, great. That's another good question. So we have data sets that span binding, HAI, and neutralization. We have found that HAI versus neutralization is incredibly tight. So we have some data sets where we plot them both. They are, for us, just y equals x. And so I mean, like truly a one-to-one ratio within the error of the HAI assay itself. So I treat those nowadays as identical. Binding turns out to be different. So surprisingly, what that means is that most of the antibody response is targeting the HAI head because that's all that HAI is measuring, and there's not a lot going to the stem, which I think is in line with what the literature shows. So the short answer is that HAI neutralization we treat as identical. We can absolutely separate them out if we need to, but the predictions just end up not mattering in that case. So we lump them all together in terms of just more predictive power.
Thank you. Then I see another question from Soho Muhammad Usman, and he says, looking at limitations, can HAI test accurately predict protection against influenza variant with immune evasion properties, which may not reflect HAI assay? And thank you for your great presentation.
Okay, thanks, Steve. So that's another excellent question. Many ways to answer that one. So ideally, what you do with the HAI assay is that you test it against the virus that you care about. So if, for example, the vaccine strain has one virus inside of it, and then there's escape that happens and another virus circulates, then you really want to measure HAI against that circulating strain, and that should be correlated with protection against that circulating strain. So it is true that if you're still like insisting on HAI against your vaccine strain, it might not be an accurate reflection of what happens here. I will also add to that the fact that if you look over the past 10 seasons, then the number of times that we get immune escape turns out to be very rare. So there were up to this past year 4 different components to the vaccine, and you can ask for each of these 4 components for each possible year. How frequently are the viruses that people are infected with? How frequently are they the strain that's in the vaccine? And the answer turns out to be 90 plus in almost every single case. So there were a few cases of escape, but really the challenge with influenza appears to be nowadays, in my mind, not exactly how to predict what's going to happen. Those predictions are fantastic. The questions are, is there going to be a big shift, like an antigenic shift, or also how can we take the normal seasonal vaccine when everything is going right and make it as good as possible for everyone? And so the weak responses that I've shown here that happen for 2/3 of people are not happening because the virus keeps evading. It's in fact staying exactly where we think it is. It's just the immunogenicity of the vaccine that we're giving doesn't appear to be that potent.
There are lots of questions. So I'm going to read you a couple more, but then I also have a question. So I'm going
Okay.
to
And
give
Sita,
my,
we also have 2 hands raised that have
okay.
been up for a while, so we can get to those as well.
Okay, so let me just read you the Michael Carlock's question, which
Mhm.
is again very nice talk. Does your model incorporate antigenic um, cartography?
Okay. The short answer there is no. We use a different method that gets at the same heart of the idea, but we use our own machine learning through random forests.
Okay, so let me ask my question, and then we
Mhm.
can go to the raise hands. So my question is, especially for this talk for influenza, the antigenic sin is a big thing,
Mhm.
and depending on who got infected or vaccinated with what vaccine strain or the infection strain, and
Mhm.
how you managed to incorporate or were you able to manage and to incorporate the original antigenic sin in your prediction. And then I'll add one more question to it and how this prediction helps us understand about creating a universal vaccine.
Okay, great question. So this is an effect that people bring up over and over again, as they do many other things. My short answer is that the 2 most well-known effects, and I think they're just well-known because they're popular, not because they're large effects, is original antigenic sin and the antibody ceiling effect. The original antigenic sin says that your age matters, and that's going to influence your antibody response. Our data suggests that your age matters, but it matters very subtly and rather a small effect. So here's like the example of children versus adults on the left here again. And you can stratify the adults by different age groups and think about the age groups corresponding to which virus they saw first. We truly do not see a large impact on this front. So we like to say that original antigenic sin is this big effect, but when it comes to predicting what you versus I versus a child versus anybody else is going to elicit, it seems to not be that big of an effect. So this is really, the most stark effect size that you can see here. It goes from a prediction of 2x to a prediction of around 2.5x on average.
Yeah.
Um, yeah, go ahead.
Thank you for that. Uh, Ted, would you allow the person who raised their hands because I
Yes,
can't
so I'll
see?
allow Chapa and Korosek. You can ask your question directly now.
Oh, so I'm unmuted now.
Mm-hmm.
Yes, we can hear you. Yep.
Thank you so much for this awesome talk. I also transitioned from physics to theoretical immunology, so I have lots of questions for you. But for
Mm-hmm.
your competition, I saw CD4 metrics that you're tracking at baseline. I'm wondering if you're also doing CD8 or CD4, CD8 ratio at baseline, because I know that's a pretty good metric for overall immune health, often used in HIV, but in other contexts as well. And my second question is, you kind of already alluded to it, you're using random forests, but I wonder for your imputation work that you talked about towards the end of your talk, how specifically are you doing that imputation algorithm? Because I know that the random forests, the way that they're capturing interdependencies across like these multi-omics data sets can matter depending on how you're doing your k-fold cross-validation or how you're splicing up people and stuff like this.
Okay, good question. I'll answer both briefly. So CD8 we are not going to measure. We think from the literature that CD4 is the dominant factor, and that's really what we're focusing on here. I think for influenza, it's going to end up being that CD4 is what matters. This is what we see from folks like Paul Thomas, who said, he was adamant that he absolutely loved CD8, and eventually he was persuaded that CD4 is really the dominant factor here. And whenever you hear something like that, and you say, okay, I think this is really what we should measure. So we're trying to get the most bang for our buck. We're staying with CD4 only. In terms of the random forest imputation, we haven't done anything yet on the slide that you see here in terms of sequencing. Absolutely, the cross-validation will be important. Everything that we've shown in this talk is really based on serology, and then coding was done very straightforwardly as each variant is encoded just using its name, which means that if you're the same virus measured in two different studies, you're the same virus. Otherwise, you're considered to be a distinct entity, and we just let the forest learn on its own. We are now looking into whether you can do something fancier using the HA sequence to predict even better, but this already turned out to be so excellent. It's like we have this preponderance of data, and so we just wallowed in the luxury of having that and just let the data tell us how each virus behaves.
Awesome. Thank you so much.
And then I wanted Sita just to allow Jeffrey Almond. He had he had
Sure.
his hand up for a while, so I wanted to give him the opportunity.
Go ahead, Jeffrey.
So Jeffrey,
I actually
awesome.
wrote my question as well, and my question relates to antibody subtypes, IgG 1, 2, 3, 4, whether there is any correlation there if people have similar subtype differentiation with the way they'll respond.
So that's a great question, and the answer there is I simply don't know, because none of our data sets that we have so far have differentiated between subtypes. We just have serum titers.
The reason I asked the question, there was a paper published last year that said that even if you don't have neutralizing antibodies, if they're of a certain subclass where you recruit ADCC, you can have a good level of protection via that mechanism, and that would be more broadly based. And it may be that the virus via NS1 immunomodulation, etc., can influence subtype differentiation. It's a subject we don't follow very closely, but it's sort of interesting that that if you can recruit ADCC, you don't need to neutralize, you just need to bind to the virus-infected cell, and off you go. Anyway, it's a thought.
I agree. I love that idea. I would say that whenever we have the data on this slide, it should be more accessible. And I would love to know if some people have weaker antibody responses but stronger T cell responses to compensate, or cytokines or what have you. It's a great question, and I would love, as soon as we get those data sets, the answers naturally flow out. At the moment, I just don't have a good answer on that front. But thanks
All
for the
right,
question.
thank you. Thank you.
Would you answer a couple more questions? It's
Sure.
2 minutes left. Okay. So John Paulo asked, some studies have found that repetitive immunization reduce the responses and avidity of antibody response. Have you observed something similar in your model?
Yes, so we have found that there is an effect of repeated vaccination. We haven't put it in yet into our model because now we're in a very niche area where you're following people year after year, or you have their vaccination history, which we don't always have. The short answer is much like on this previous slide, we have seen this effect and it is small. That's kind of the moral of the story with most things in influenza. Again, the largest one turns out to be when the study was conducted and wasn't more than 10 years apart. Repeated vaccination history is one of those things that you will only see the effect when you have enough individuals. It's never going to be some huge, your response goes from 64x fold to 4x fold.
Yeah. And John Betts have asked, within your CD4 positive analysis, are you looking also specifically at CD4 Tregs?
Okay, again, good question. None of that T cell analysis has been done yet. Are we going to look for Tregs? The answer will be that we need to think about it. We'll get back to you as we finalize all these details in the next coming months and all the experiments are coming up. It's a good question about Tregs.
Okay, so we are right at 12. So
Can we get in one more from one of our
can
next
we?
gen?
Okay, Scott Snyder. If pre-infection is a factor, do mice and ferret resembles predictive value as you see in the child?
Oh, another excellent question. We don't have great data on that yet because we don't have a lot of children data. But that's the next question that we're asking because you would imagine that pre-infected should be the most closely similar in terms of infection history to children. We're looking into this. Would love to have an answer very soon.
Okay. Oh, there's.
And we have one more from Omalara, who's part of our next gen group. So
Go ahead. Oh, she's mute. You're mute.
okay, well, Omalara, you can send your question in, and we'll make sure it gets to Professor Einav.
Okay. So I think we went 1 minute over, and thank you so much, Tal, for answering all the questions. This
you all.
was amazingly interesting talk for all the audience, and thank you so much for your time and patience.
Thank you, Sia. Thank you, Ted. Thank you all. Happy to answer any questions. Feel free to reach out.
Bye-bye.